Yeah.
Elizabeth, you're free to start.
So hello everyone. I may not say good morning or good evening because we have different time zones. So welcome to the oral abstract session on moving away from isoniazid for TB preventive treatment. So I will be one of your moderators for this session. I'm Elizabeth Cadena from the Philippine Tuberculosis Society and currently the Vice Chairman of the Asian Pacific region. Before we introduce our our speakers, may I introduce my co-moderator, Neil?
Yes, my name is Neil Martinson. I work in South Africa, in Soweto, at the Perinatal HIV Research Unit, affiliated to the University of Witwatersrand. Thanks. Back to you, Elizabeth.
Okay, thank you, Neil. So may I introduce to you the first three presenters for this session. Omri Arbiv from the University of British Columbia, who will be presenting a study on safety of high-dose rifamycin for active and latent TB, a systematic review. Next would be Budi O. Susanto from Uppsala University. Her paper is entitled Rifampicin Maximum Early Bactericidal Activity Not Reached at 50 micrograms per kilogram. The third one would be Eugenia Mwamba from the Center of Infectious Diseases and Researches in Zambia. The paper is entitled Treatment Outcome of People Living with HIV on TB Preventive Therapy in Lusaka, Zambia. Back to you, Neil.
Yes, and the people who are I'm going to be introducing are Dr. Jan Yan Wang, who is from National Taiwan University and is a clinical professor in internal medicine. And the second speaker will be Karen Turner from the Aurum Institute, and she has worked extensively on the Impact for TB project, scaling up 3HP. The third person is Tay Tu Thi Dong, who has extensive experience in implementing public health programs, and he'll be discussing latent TB infection and treatment using shortened treatment regimens. And the final speaker for this afternoon will be Hung Ling Huang, who will be talking on the impact of age on completion rates and systemic drug reactions of rifapentine-based weekly therapy for latent TB. And he is a pulmonologist at Khoa Sing Medical University in Taiwan and is an infectious diseases person. So thank you very much, and back to you, Elizabeth.
Okay, thank you, Neil. So maybe we can start the presentation of the papers now.
Hi, everyone. My name is Omri Arbiv, and I'll be speaking to you about the safety of high-dose rifamycins for active tuberculosis. This is research done under the supervision of Dr. Johnston in collaboration with the McGill International TB Center, the BC CDC, and the University of British Columbia. I have no conflict of interest to declare. And so rifamycins, rifampin, rifapentin, and rifabutin are really the backbone of tuberculosis therapy. However, there is growing concern that the dosing that we're using is inappropriate for our patients. The original dosing was really based on the minimal effective dose rather than the ideal dose, as well as price and toxicity. But now we wonder, can we safely provide rifamycins at higher doses for our patients? And so to answer this question, we performed a systematic review and meta-analysis. We included all humans with tuberculosis, whether it was latent or active. And within active, we included pulmonary, TB meningitis, and all forms of TB. for patients of any age. We wanted to trials that compared high-dose rifamycin to standard-dose rifamycin, and we included RCTs and prospective cohort studies. We included studies that were published between 1965 to 2019, and we excluded trials with retrospective collection of safety and efficacy data, as well as large loss to follow-up. In terms of definitions for the study, we defined a severe adverse event as a CTCAE grades 3 to 4, and in one instance, when that was not available, we used the D8 score, and again, grade 3 to 4. In cases where neither of these were available, we used just adverse events requiring cessation of therapy as our definition for severe adverse events. In terms of high-dose rifamycin, we included rifampin greater than 600 milligrams daily or greater than 10 milligrams per kilograms daily. Anything less than this for rifampin, and anything less than this was counted as standard dose rifamycin. In terms of our primary outcome, our main outcome was total severe adverse events. In terms of secondary outcomes, we divided this by safety and efficacy outcomes. And within safety, we looked at death, severe adverse event by organ system, study withdrawal, severe adverse events in studies with low risk of bias. And we counted this as based on the ROB 2.0 by the Cochrane group for RCTs and the Robbins I for cohort studies. We also looked at severe adverse events for rifampin studies only, as there's the most prevalent rifamycin. Severe adverse events that was possibly, probably, or likely therapy related. And in terms of efficacy, we looked at relapse and 2-month culture conversion as our efficacy secondary outcomes. Moving on to results, we identified 7,800 articles, and of those, 12 were included in our systematic review and meta-analysis, 10 of those being RCTs. All studies were of active TB, and only one trial was of children. Within this, the majority of the trials were of pulmonary TB, and we ultimately included over 3,000 patients with TB for just under 4,000 person years. This is our primary outcome. So this is a forest plot looking at the incident rate ratios for total severe adverse events by each of the studies. On the right, to the right of the standard line at 1, you see that there are more severe, is pointing more towards more severe adverse events in the high dose rifamycin group. And in the left, more severe adverse events in the standard dose rifamycin group. In total, you can see that although it is trending more towards more severe adverse events in the high dose rifamycin group, it is not statistically significant. So there are no differences between the two groups with regards to total severe adverse events. This was a null result. Here, we are not looking at, this is some of our secondary safety outcomes, and we're not looking at a forest plot, but rather we are looking at the total incident rate ratio for each of these outcomes on the left. You can see here that the majority of these, again, are also not statistically significant. And I do want to draw your attention, however, to the left here, to severe adverse events by organs. We did divide severe adverse events by hepatic abnormalities, GI, hematologic, and cutaneous, as these were the ones that we associated with rifamycin toxicity. We included all other organ categories in the other organ category here below, and you can see that the other organ category, there were more severe adverse events in the other organ category in the high dose rifamycin group compared to the standard dose rifamycin group. All the other incident ratios here were not statistically significant. Here we're looking at other safety outcomes, and here we're looking at odds ratio for death and withdrawal from study. And as you can see here, both of these, this is a forest plot for both, and both of these were not statistically significant. If anything, there was perhaps more death in the standard dose rifamycin, but again, this was not statistically significant. Here we move on to efficacy outcomes, and we are again looking at odds ratios and forest plot for relapse and culture negativity at 2 months, and you can see that both of these are also not statistically significant. And so we ultimately conclude that there are no changes in severe adverse events between high dose rifamycin to standard dose rifamycin. We also note that the only difference that we noted was in the other organ severe adverse events category. And we can think of a few reasons for this. One, it could be that rifamycin just lead to different severe adverse events at higher doses. But it could also be that this is a spurious result. When we removed one study that accounted for a large amount of the weight in the overall systematic review and meta-analysis, this result would no longer held. So we do wonder if it's just driven by one trial. The other thing we wonder is if this is potentially because of earlier studies that were unblinded and had the investigators stop rifamycin at the earliest sign of any toxicity, and maybe that led to more severe adverse events. We do note, however, that there are no differences in the outcome of death, which is the most important outcome when we're thinking about our patients. But we also do note that there are similar efficacy outcomes with regards to relapse and 2-month culture conversion with high dose to standard dose rifamycin. This is in line with similar reports. So in 2011, Steingart et al. Performed a systematic review and meta-analysis of just high dose rifampin and found similar results. And this is reassuring with regards to our findings as well. And I guess this leads to our final point, which is while we haven't identified a clear benefit of high-dose rifamycin, certainly we think that future studies should try to see if there are certain subgroups that do derive benefit from high-dose rifamycin, and perhaps that's where future work should go. And with that, I conclude my presentation. Thank you so much.
Okay, hello everyone. So I'm Putri Oktasari Susanto from Uppsala University, Sweden, and I'm presenting also on behalf of Panacea Consortium. And today I would like to present about our modeling analysis on high dose of rifampicin trial, which shows that rifampicin maximum early bactericidal activity is not reached at the 50 milligram per kilo. In this presentation, I declare that I have no conflict of interest to report. So the current standard dose of rifampicin, which is 10 milligram per kilo, has been used for decades, although this dose was chosen not based on the dose ranging trial, but mostly because of the economic reason, fears of toxicity, and pharmacokinetic considerations. And several studies had suggested that 10 milligram per kilo of rifampicin is suboptimal. And one of the trials evaluating the high dose of rifampicin is Panacea HiRIF1 trial, where they evaluated rifampicin dose at 10 to 15 milligram per kilo. So the patient was treated for 2 weeks. On the first week, the patient was given rifampicin in monotherapy. And on the second week, the patient was treated with rifampicin in combination with the other drug in the standard dose. And for day 7 and day 14, the plasma concentration was taken for pharmacokinetic assessment, as well as daily sputum collection for early bactericidal activity assessment. So there are some results on the 10 to 14 milligram per kilo that has been published. So the first publication is where they developed a rifampicin pharmacokinetic model, where they are able to describe the area under the curve of rifampicin at different doses at day 7 and day 14 using the model. And then the other publication where they develop a pharmacokinetic and time to positivity model in order to describe the exposure-response relationship, as shown in the plot where they relate the dose with the change from baseline time to positivity at 10 to 40 milligram. And they even able to simulate and predict how the response for dose 45 and 50 milligram. So since we received the data for 50 milligram, and then the objective of our study is to evaluate if a maximum early bactericidal activity has been reached at rifampicin 50 milligram per kilo. We did this by investigating if earlier developed rifampicin pharmacokinetic and time to positivity models are applicable at the 50 milligram per kilo dose. So we started from the data that we received from Panacea HiRIF1 trial, where the patient received rifampicin from 10 to 50 milligram. And for the plasma concentration time profile, we analyzed it using the earlier developed rifampicin population pharmacokinetic model. to see if the new data can be described with the model. And for the sputum sample that has grown in the liquid culture until it gives the positive signal, we call it time to positivity, and we use this data to and analyze it using the rifampicin time to positivity model to also see if the model can still describe the new data. So for the pharmacokinetic, it shows that the earlier developed rifampicin population pharmacokinetic was able to predict or describe the 50 milligram per kilo data. As you can see in the plot, that the model predicted AUC was in agreement with the observed AUC at day 7 for different rifampicin doses, including 50 milligram per kilo group. And I also want to highlight some of the key features for the rifampicin pharmacokinetic model, which are including dose-dependent bioavailability, the enzyme autoinduction, where rifampicin induced the enzyme that is responsible for its own metabolism, and then nonlinear eliminations. For the time to positivity, the earlier model was also able to predict 50 milligram per kilo time to positivity observation. As you can see in the red shaded area is the model prediction, where the black solid line is the observed data, and it's still within the range of the model prediction. We can also see that there is still an increase in the time to positivity of dose 50 milligram compared to 40 milligram, which means that the maximum early bactericidal activity or the efficacy has not been reached at 50 milligram per kilo. But there are some potential tolerability problems, which described in this publication, where the patient with a higher dose had higher probability of experiencing tolerability at first event. As you can see in the plot for the dose 50 milligram on the x-axis, the probability of the patient experiencing different number of tolerability at first event is higher compared to the other dose group. So what I conclude from my slide is that the earlier simulation of rifampicin pharmacokinetic and time to positivity after 50 milligram per kilo were confirmed. Even though we see that rifampicin pharmacokinetic was nonlinear, but the exposure-response relationship in this case related to the time to positivity is still linear for 10 to 50 milligram per kilo, which means that the maximum early bactericidal activity is not reached at 50 milligram per kilo. And one important point that needs to consider is the clinical use of rifampicin might be limited by tolerability at 50 milligram per kilo. I would like to thank our colleague, our partner and collaborator, who involved in this study for all of the contribution. Thank you.
The 51st Union Conference, the 51st Union World Conference on Lung Health, Advancing Prevention. I have no conflict of interest disclosure. My presentation is on treatment outcome of people living with HIV on TB preventive therapy in Lusaka, Zambia. This presentation is by Eugenia Mwamba. Background: TB mortality among people living with HIV is over 2.5 times higher than the HIV negative population. 59% of the reported TB cases are TB HIV co-infected. TB preventive therapy, TPT, reduces the incidences of TB among people living with HIV. The reduction in TB incidences is more significant in patients who complete a course of TPT compared to those that do not. Uptake of TPT has been lower in Zambia and other high burden settings due to fear of adverse outcomes, fear of increasing drug-resistant TB, represent the treatment outcome of people living with HIV who were initiated on TPT in Lusaka District Health Facilities in Zambia. Methodology. A retrospective analysis of IPT program data between October 2016 and March 2020 from CDC and PEPFAR-supported facilities under the ACHIEVE program was done. So here we have 158,020 who were eligible on treatment. This was our eligible treatment curve. And then for those that were initiated on TPT, we had 54,272, which is 34%. And then those that completed were 42,940, that was 97.1%. And those that died, there were 36, which was 0.1%. And those that were lost to follow-up were 1,625, which is 3%. And those that developed TB were 146, which was 0.3%. Then of those, that were initiated on TPT, of those initiated on TPT, there were 34,279 initiated on TPT and 26,985 completed. And then we had those were females. And then for the males, we had 19,993 males, and those that completed, 19,993 were initiated, and 15,955 completed, which was 79.8%. Then conclusion, much as TPT initiations are still low, TPT completion rates are high across both genders and age groups. Less than 5% of people initiated on TPT had adverse outcomes, suggesting that TPT is safer than it is presumed to be. Improving uptake of TPT among people living with HIV is urgently required in order to reduce the TB burden in this high-risk group. Those are my references. Thank you.
I thank the organizing committee for giving me this opportunity to present the preliminary results of our randomized control trial comparing two rifampicin-based short course regimen for the treatment of latent tuberculosis infection. I have no conflict of interest relevant to this presentation. The figure nicely illustrated the importance of intervention against latent tuberculosis infection for TB elimination by 2050. By treating active tuberculosis, preventing infection, and mitigating risk factors, we could possibly reduce TB burden significantly, but never eliminate it. We can only achieve that by treating active tuberculosis as well as latent tuberculosis infection. Treatment for latent tuberculosis infection, however, is never easy. The completion rate is far from satisfactory. Fortunately, in 2011, a breakthrough in preventive therapy happened. With rifampicin-based modern short course regimen, so-called 3HP, the completion rate of preventive therapy increases to more than 80%. Yet, the risk of adverse drug reaction under 3-HP is significantly higher than conventional 9-month isoniazid regimen. Under such a high unit dose, 15 mg per kilo, adverse drug reaction due to 3-HP is not uncommon, especially in subjects between 35 and 65 years old. In this age group, the risk of systemic drug reaction can exceed 15%, most of them being severe flu-like symptoms. By using a smaller unit dose, 5 mg per kilo for isoniazid and 10 mg per kilo for rifampicin, and giving on a daily basis for 1 month, results of the brief TB trial shows that 1-HP was non-inferior to 9-month isoniazid for preventing tuberculosis in people living with HIV. The completion rate was significantly higher in the 1-HP group. In addition, the rates of combined grade 3 and 4 serious adverse events and targeted safety events during treatment course was lower in the 1-HP group. So based on the safety profiles in the preventive trial and the brief TB trial, we hypothesize that the risk of systemic drug reactions under 1-HP will be lower than that under 3-HP, leading to a further high completion rate for preventive therapy. Therefore, last year, we began a multi-center randomized controlled trial on non-HIV close contacts with latent tuberculosis infection to compare the risk of systemic drug reaction. The study is supported by Taiwan CDC and the Ministry of Science and Technology. It is conducted in 6 hospitals in Taiwan, including 2 medical centers. Close contact is defined as staying with an infectious index case in the same ventilation unit for more than 8 hours a day, or cumulatively more than 40 hours. Latent tuberculosis infection is diagnosed by either positive quantiferon or positive tuberculin skin test. In addition to the incidence rate of systemic drug reaction, we evaluate the completion rate, safety profile, and risk of developing systemic drug reaction. The number needed was estimated to be 245 each arm. The first case enrolled in last September up to this January. A total of 172 latent tuberculosis infection case was screened for eligibility, and 129 cases were enrolled. 69 and 60 cases were randomized into the 1-HP and 3-HP groups, respectively. The completion rate in both groups was above 90%. 13% of the 1-HP group and 15% of the 3-HP group suffered from systemic drug reaction. However, the systemic drug reaction was predominantly flu-like symptoms in the 3-HP group, but predominantly urticaria in the 1-HP group. Okay. Sorry, back. Probably because of small number of cases, the 2 groups was different in sex and smoking status. The mean age was slightly above 50 in both groups. Hypertension and diabetes were very common in both groups. Though all study subjects were quantiferon positive, the interferon gamma response was significantly higher in the 1-HP group. Other laboratory results and the characteristics of exposure were similar between the 2 groups. This slide shows the details of adverse drug reactions. Though the risk of systemic drug reaction was similar in both groups, 13% versus 15%, clearly 1-HP had a high risk of cutaneous reaction, with 8 experiencing urticaria. 3-HP had a higher risk of flu-like symptoms, especially fever, nausea, and vomiting. The most common cutaneous reaction was maculopapular eruptions, which was not very itching and tended to be improved by antihistamine. PK analysis failed to show any associations between the C2 and C6 plasma drug concentration and the development of urticaria. In conclusion, the preliminary results shows that in non-HIV close contact with latent tuberculosis infection, completion rate and the systemic drug reaction risk was similar in 1-HP and 3-HP groups. However, the side effect profile was quite different, with more cutaneous reactions under 1-HP and more flu-like symptoms under 3-HP. And cutaneous reactions seems have nothing to do with plasma level of isoniazid and rifampicin. Thank you for your attention.
Hi, I'm Karen Turner with the Orem Institute and the director of the Impact for TB project. I have no conflict of interest to report for this session. And today we're going to be talking about preparing for short course regimens in countries and how we did it at Orem through the Impact for TB project with our partners. So first I want to begin with what is 3-HP, just a little bit of an overview. It's a new short course regimen that's been available for some time on the market, but not for high burden TB countries. WHO guidelines updated it in 2017, 2018 to include 3-HP, and now we also have 1-HP included for adults. Why is it a game changer? It's shorter, it's less toxic, safe to take with both dolutegravir and efavirenz-based ART regimens. It's less burden to the health system for many reasons that we'll get into, and it has similar efficacy of other TPT regimens. So as a product overview, there's 2 versions available now. We have the version that's been around for a bit, Sanofi Prifton tablets that are combined with INH tablets. You take those once a week for 3 months over 12 weeks, and you take the same pills in the same 12-week period for the FDC, but it's only 3 pills. And as you can see here on the dosage of the 3-HP medication, there are 6 red pills of Prifton represented here that you combine with an INH. That is 1 dose for a patient to take. And as the FDC comes into market very, very soon, once it clears WHO QA, you're going to be taking just 3 tablets once a week. We do add on pyridoxine at 25 milligrams for people living with HIV. Another thing to note is the child patients are not able to take the FDC right now. That is only for an adult formulation, but what we do for children 2 to 14 years is take the rifampicin tablets that are Prifton and crush them to give to kids. There is not a dispersible version yet, but we'll explain that a little bit more. So what we looked at in this abstract is what were some of the obstacles to adoption, and also to help other countries, if you're in the audience too, of how you get your country ready to adopt 3-HP or similar short course regimens. So in the beginning, when we started the grant in 2017, rifampicin just had a single supplier, Sanofi. It was very expensive. Even before the project began, it was $72 a patient course in the US market. We got it down to $45 with work with GDF. And then we got it further down, as we'll talk to in a minute. We weren't sure if rifampicin could be safely used with dolutegravir, and that was a critical focus for countries that were transitioning off of the efavirenz regimens over to the dolutegravir regimens. And we knew, as I showed on the other slide, the pill burden of 3-HP was still a little high. It's a lot of tablets to take at once with the Sanofi Prifton tablets, and then you have a box of the Sanofi tablets, and then you have a box of the INH that you must combine somewhere along your supply chain system. It was also not really included in national guidelines in 2017. WHO issued it globally, and then gradually been working to get them in the local guidance in countries. And we also knew that it couldn't be given to children under 2. We didn't know the dosing for children and if it was safe, and we also didn't know the same thing for pregnant women. The Impact for TB project, which stands there on the acronym, started in 2017 with a significant investment by our donor, Unitaid. The consortium is led by Orem Institute in partnership with KNCV, Johns Hopkins, and Clinton in countries and at the global level. TAG is also our partner in working with civil society organizations across our 12 countries, and we have a collaborative relationship with GDF and the World Health Organization. We are in these countries, 12 listed below, both high middle income, low income countries. There was a nice match with some of these countries, as you know and can see, for those getting Global Fund grants and PEPFAR, as we'll talk to in a bit about how those investments came to the fore as well. So the project is $60 million over 4 years, but we're hoping to get an extension on that. We had a slotting amount of about 18 million for procurement of the product. That could be the product that is the FDC, the Sanofi singles, and potential child-friendly formulations if those come to market. The focus of the project really is on people living with HIV and children under 5, although in many countries we've had to adopt that because of the guidance being 0 to 15 for children. And we knew that we were going to be working in 3 different quadrants, working at market interventions at a global level, supporting the delivery at a country level, and then across selective countries we'll be generating evidence of how 3HP rolled out and the impact of 3HP rollout. So what have we been doing up until now, October 2020? So out with old, in with the new. Rifapentine now has 2 suppliers. We have a generic supplier, McLeod's, providing the FDC. A third is on the way with a potential 300 milligram single tablet. Rifapentine is more affordable. It went down from $45 to $15, although it's still slightly more expensive than IPT. Rifapentine is shown now to be safe to use with dolutegravir, thanks to the Dolphin trial that Orem led with Johns Hopkins University. The FDC has lowered the pill burden, as shown in the diagram below, or before, and then the study of 3HP in young children, TPTC35, which we are collaboratively supporting with CDC and through Stellenbosch, has started. Hopefully those results will be in sometime next year on a fixed dose combination dispersible tablet for children. So we will now know for the little guys, the 0s to 2s, if it's safe and how you can dose 3HP. A study is ongoing with 3HP in pregnant women. It looked pretty good in the initial results for the impact on pregnant women, but we do know that the results for what it does to the fetus are still not known clearly, and so most countries still do not recommend 3HP for pregnant women. And as part of our studies, we're also looking at the cost effectiveness as part of the result of the Impact for TB project. So in the beginning, rifapentine really was not on any essential medicines list at all, and right now we see 4 out of 12 countries with rifapentine registered and approved under their national drug authorities. We have rifapentine on an essential medicine list only in Kenya and Zimbabwe, and this limitation largely has to do to price. I think the $15 price is still seen as slightly more expensive, so the cost effectiveness work that we're going to be looking at under the project will really help boost the amount from 2 to 12, hopefully, in the next few years. Also a key milestone, we had to get in the national guidelines, and there's not only one set of national guidelines, we had to get it in the HIV guidelines, the TB guidelines, and the maternal child health guidelines due to the populations that most benefit from 3HP and were targeted under this project. Right now, we see HIV adopting it quite quickly. This is, as we'll talk to, an investment by donors overall in people living with HIV going on 3HP. The TB program and household contacts is up and coming, especially for adults. The children is still a little bit of a tricky area, and we have a lot of children that are opting for 3RH until the 3HP is available in a child-friendly formulation, which is impacting the MCH guidance. For overall uptake, orders on 3HP are out. We have 11 out of our 12 countries having orders. 3HP has arrived in 7 of them. We'll start delivering the product in about 9 of them. The reasons for some of the delays is COVID, as many people are going to mention in their presentations. India and Brazil had a demonstration study and was needed to have a protocol in place in order to implement 3HP. And Tanzania is still pending some negotiations at the national government level for the buy-in for 3HP and if it is a valuable regimen for that country. But do note that the delays overall we're seeing in the project have shifted significantly since 2017, and we're hoping that an extension will help us implement all the elements of our project before the end. Another significant area is the buy of 3HP. So we had started in the beginning as Unitaid with an amount of money to purchase it. You can see that starting in our countries that are in green. The hash marks are showing PEPFAR buy-in. So Namibia has already started with PEPFAR funding, and Vietnam. Then we're moving into second-tier countries that have orders but haven't started patients yet, both from a Unitaid investment and a PEPFAR investment, moving into third-tier and even fourth-tier countries. A lot of the fourth-tier countries we're seeing are maybe ongoing PEPFAR investments, but for sure the Global Fund grants are picking up a lot of the 3HP. So I'd like to thank all the contributors to this presentation. We have Unitaid being our general support for all the project Impact for TB alongside Orem, Kane CV, Hopkins, and CHI and TAG. But I'd also like to acknowledge our national TB and HIV programs as they provided a lot of the data that was required for this presentation. So many thanks, and I hope you learned something. Thanks, bye.
Hi everyone, my name is Thủy Đồng. I come from Friends for International Tuberculosis Relief Organization in Vietnam. Today, I would like to present the study Our study is latent tuberculosis infection testing and treatment using shortened treatment measurement for contact and secondary school children in Dosen District, Hải Phòng City, Vietnam. I have no conflict of interest to report. Firstly, I will talk about some TB situation of Vietnam. Vietnam ranks 11 among the 30 high tuberculosis burden countries with an estimated incidence of 174,000 TB patients and over 13,000 TB-related deaths in 2018. In order to reach the goal of ending TB by 2030, Vietnam needs to expand proactive activities to detect and treat TB cases in the community as well as expand secondary prevention efforts to identify and treat latent TB infection. And this slide, I will talk about our intervention we conducted as part of the TB Risk Funded SWIFT TB project. A TB and LTBI screening campaign was conducted in Dosen District, Hải Phòng City, Vietnam in September 2019. Quantiferon TB Gold Plus was used to determine LTBI status in 1,001 resident family contacts and secondary school students with age from 10 to 14. And in this study, the shortened 3SR measurement was used to treat eligible persons with LTBI. Local community health officers and community health workers supported the provisions of LTBI treatment, which included weekly adverse event monitoring. We have some results from our project. The LTBI rates were 19% and increased with age. In children less than 15 years, the rate was 6.3%. In participants aged from 15 to 54 years was around 36%. The age equal and more than 55 years, the rates were 58%. And the other results, LTBI prevalence in household contact was 38.6% compared to 15% for all other groups. Of the 191 people with LTBI, 157 were eligible for LTBI treatment and 77% began treatment. Nobody experienced a serious adverse event. The rate of adverse events was 17.4%, most commonly consisting of fatigue. Of those starting 3SR treatment, 83.5% of those completed the treatment, 90.9% was lost follow-up, 6.6% discontinued treatment due to side effects. From our study, we have some conclusions. LTBI treatment with 3SR were well tolerated and yields high completion rate. Needs to expand screening and early LTBI treatment. Shortened measurement may help to increase uptake and adherence. Lastly, I want to say thank you to National TB Control Program of Vietnam, Hải Phòng People Committee, Hải Phòng Department of Health, Hải Phòng Lung Hospital, Dosen District Health Center, IDVN, Friend for International TB Relief, U.S. Center for Disease Control and Prevention, Clinton Health Access Initiative Vietnam. This work was generously supported by a grant from the Stop TB Partnership TB Risk Initiative with funding from the Government of Canada.
Hello, everyone. I'm Honglin Huang, come from Kaohsiung Medical University Hospital in Taiwan. I would like to share our study online to you all. My topic is impact of age on completion rate and the systemic drug reaction of rifampicin-based weekly therapy for latent TB infection. I have nothing to declare. Treatment of latent TB infection is a keystone of TB elimination by 2035. The latent TB treatment regimen evolved for many years, and the short-term regimen containing 12 doses of weekly rifampicin and isoniazid named 3HP has gained popularity in recent years. Several advantages have been reported. When comparing to traditional 9-month isoniazid monotherapy, it had higher completion rate and less hepatotoxicity. However, unpredictable systemic drug reaction occurred in 4 to 10% among 3HP users. Before widespread use of the 3HP for latent TB infection treatment in all age population, comprehensive assessment for the 3HP safety issue is very essential. From the preventive trial, we have known that age greater than 35 years is a risk factor for SDR. However, the comprehensive study in elders greater than 65 years is lacking. We conduct this prospective multicenter observational study. We enrolled the latent TB infection population, including close TB contact and the high-risk population, such as the long-term facility residents, workers, and the poor diabetic control patients during September 2014 to December 2019. All of them received at least 1 dose of 3HP under daily observational therapy. We divided 3 groups by age. Those under 35 years are in younger group. Age between 35 to 65 years are in the middle age group, and the age greater than 65 years are in elderly group. Programmatic adverse drug reaction follow-up and management were administered for all participants. The endpoint is to compare the treatment outcomes, including the treatment completion rate and the risk of SDR in different age groups. We finally enrolled 579 cases for further analysis. The ratio of males and females were similar, and around 25% among them were elderly. The TB close contact accounts more than 75%. Considering the baseline characteristics of 3 groups, the elderly group has a higher proportion of systemic comorbidities and more radiographic abnormalities, except for TB. Here we come to treatment outcome after stratifying the age. When compared to the other 2 groups, the younger age group had highest completion rate, but we can see that half of them ever had tolerable adverse drug reactions. The elder group had higher permanent discontinuation rate, and the hepatotoxicity and the other adverse drug reactions, except for systemic drug reaction, accounts for the majority cause for termination. The systemic drug reaction is a major cause of the middle age-related termination. The details of ADRs among different groups are different. We can see the middle age group had more systemic drug reactions, especially the flu-like syndrome and urticaria. The elder group experiments more uncontrolled hypertension than other groups, and it also accounts for the majority of severe adverse drug reactions in this group. Here we come to the multivariate analysis results. Compared to the elderly group, the middle-aged group patients were significantly associated with increased SDR risk during 3HP, even in most clinical settings. The risk of systemic drug reaction was not different between the elderly and the younger groups. Okay. And the risk of treatment discontinuation was similar in the middle-aged and elderly groups. We can see the overall rates were almost similar. In comparison with the elderly group, the younger group had slightly but non-significantly lower risk of treatment discontinuation. The subgroup analysis demonstrated the consistent finding in most clinical settings. We can conclude that under programmatic follow-up and the proper medical support, the overall completion rate of the HP treatment for latent TB infection is very excellent, even for the elderly group. Current study demonstrated that adverse drug reactions monitored should also be under age stratified. During HP treatment, higher incidence of systemic drug reaction should be in caution for the middle-aged group. It always occurred at the fourth dose and lasting for 1 to 2 days. Uncontrolled hypertension for the elderly might be a big concern, particularly in those with underlying hypertension. Most of them can complete 3HP after modification of antihypertension drugs during the 3HP treatment course. And it's all, and thank you for your attention. And I also want to thank for all sponsors and the partners in Taiwan who helped us to complete this study. Thank you. Okay.
Okay. Can you hear me?
Yes, we can.
Okay. So thank you to all the presenters. It was good work. Yeah. We have some questions here and we are given a few minutes to answer this question. So may I, the first question is, I think, for Omri Arbiv. The question is, could you please provide examples of civil adverse events belonging to the other organ category?
Sure thing. Yeah, happy to provide that. Thanks again for having me in this conference. In terms of the other organ category, so we divided this. We categorized it as the same as the CTCAE criteria or the CTCAE adverse organ categories. And when going by those, the most common severe adverse event in the other category was a nervous system disorder, and that accounted for more than 50% of the severe adverse events. We think this is largely driven by a very large TB meningitis trial that accounted for most of the severe adverse events. There's the other three common ones were investigations that were abnormal, so lab results for the most part, respiratory disorders, and the last one was immune system disorders as well.
So there are a couple of questions for Karen. The first one that comes up is is there evidence that rifapentine can now be used with dolutegravir in antiretroviral naive patients? Would you like to just comment on that?
Sure thing. Yeah, sorry I didn't go into detail of all the studies. So Orm had started with the Dolphin trial, which showed that 3-HP and dolutegravir is dosed and safe with not treatment new. So we were asked by PEPFAR and WHO to extend an arm into the trial to look at treatment naive patients. So we have been ready to go with that trial. There are some delays. I see another question in that regards to the product rollout in the country of South Africa, which is limiting when we can start, but we're ready to go. And that study will take about 12 months and we'll be able to show also in treatment naive if it's safe. But so far, so good. We have about half of our countries that have accepted the evidence that came from Dolphin and do not require this additional evidence to initiate patients on dolutegravir and 3-HP. Over.
Thank you, Karen. And then there's another one while you have the floor is about nitrosamine and the impurities and how they impacted on the rollout of rifapentine containing regimens.
Yeah, I kind of knew this one would come up here. So I'll give you what I know. And I think officially there'll be some words that are going out to all countries around this issue. So most people have seen the Sanofi reports that have come out where the FDA became aware of a nitrosamine impurity that was in rifapentine. It's in the API. And they had done their due diligence as a manufacturer to figure out what was the parts per million threshold that would be required to be safe for human consumption. That also went through WHO and the Global Fund, and they issued a notice to countries around that threshold being below 14 parts per million. They have done some further analysis, and we're hearing that it will actually rise a little bit. That was a very conservative level. So the Sanofi singles may be coming forward to your country soon, but they're still on hold for distribution and must clear these tests to ensure that the parts per million is below the advised threshold. So if there's a slight amendment in that, I think it might be going a little bit higher. You still require that before you get the product in your country. The FTC, it's not known yet whether the production of the API also has cleared this limited threshold. It is going through drug authorities, both in the US and in Europe, and WHO will clear that. And then we'll have that pending outcome, hopefully by the end of this year for both products. And for children, maybe Neil, there is a definite stop in the manufacturing of the child-friendly formulation that I mentioned was under TBTC 35. They also need to get the threshold down. It's a different threshold for children than it is for adults. We're not sure about the timeline for that, but that will be further delayed because it needs additional studies just for the child-friendly formulation. Over.
Thank you very much.
Okay, thank you. For Eugenia Mwamba, what TPT regimen was used in your study?
Okay.
Hello.
Yes.
We used isoniazid.
Okay, thank you. So there are also several questions for Dr. Jan Yuan Wang. Okay, in the 3-HP versus 1-HP, the number of the denials were high. Can you talk about reasons? Is it the same with 6 months of INH?
I'm sorry, could you repeat the questions?
I think the question is the denial is very high. Can you talk about the reasons? What are the reasons why? 4, 4, 4.
What is denied? I'm sorry. What? Can you type the question for me, or?
Yes, it was addressed to you.
Addressed to me. Okay. Questions.
how many patients were dropped off from this study?
Dropped from this study.
Yeah.
But I don't exactly know the questions, but you can see the completion rate is quite high. It's about 90% completion the either 3 HP or 1 HP regimen. The completion rate is very high. So it's
meaning to say maybe they, yeah. Yeah, in the 3 HP, it's 82%, it's 32% completion, right?
Yes. In the 1 HP group, the completion rate is 93%.
90% versus 82 for the 3 HP.
I'm sorry, you mean the side effect, the
completion.
The risk of, no, the completion rate is sorry.
So I think they are asking the reasons why, like in 82%, the reasons why they were not able to complete the.
For the short regimens, I think the most important reason for not completion is the side effect, because the regimen has already very simple and easily to implement in Taiwan. As I said in my slide, that the risk of systemic drug reaction is slightly higher than than we previously think, and predominantly in the 3 HP group, that is flu-like symptoms, and in the 1 HP group, it is urticaria and other cutaneous reaction.
So that is the common reason. Now, just a personal question regarding the cutaneous side effect. Is this something temporary?
Of course, it's temporary, and usually can be easily managed by either topical or systemic antihistamine.
Okay. Neil?
And then Dr.
Any other
Wang,
question?
Yes, another one for you, Dr. Wang. There's a question about the timing of the adverse events. Did most of the events tend towards occurring after 1, 2, 3, or 4 doses?
For the 3 HP group, the most common side effect happens at the third and the fourth dose of 3 HP. But in the 1 HP group, most cutaneous reaction develop after 7 to 10 days after treatment. 7 to 10 days after treatment in the 1 HP group.
Thank you. And then a question for Dr. Dong. What proportion of people with LTBI were initiated on 3 HP or any other regimen? Dr. Dong, are you there?
Oh, yes. Sorry. Can you repeat your answer again?
Yes, of course. The question relates to the proportion of people in your study who were initiated on preventive treatment, and I think people are asking what proportion of people were initiated on 3 HP.
Oh, yes. In this study, we did not use 3 HP because so all of those patients were initiated on 3SR.
So no patients were initiated on 3HP.
And yes, and the proportion was 77.1% began to react at treatment.
Thank you.
Yes, thank you.
Are there any more questions, Neil, that we may have missed?
Yeah, so I have a question for Omri Arbiv, just about, he mentioned there might be some instances where high-dose rifampicins may be of value, and I was just wondering what are those instances where these high doses may be of assistance, especially when this data sort of shows quite a similar adverse event profile and efficacy profile.
Yeah, so that's a great question. So I guess so in terms of our specific study, as you were mentioning, just did not demonstrate any benefit of using high dose rifamycins. What we are thinking though is we did include all patients who were treated with high-dose rifamycins with no exclusion of any age, comorbidities, et cetera. But we do wonder if maybe high-dose rifamycins are not the best treatment for all comers, but maybe there are specific subpopulations, perhaps patients who are younger or have fewer comorbidities who might be able to benefit from high-dose rifamycins.
And the other question I just wanted to find out is you sort of alluded to this in your talk, but it was a bit unclear as to how many of these studies were placebo-controlled or had some design device that prevented the investigators from knowing who was getting high dose and who wasn't. Because I suspect that investigators may be more cautious if they know that the participant is receiving high dose.
Mm-hmm. That's a great question as well. So all studies were selected to have at least one arm that was standard dose rifamycins and one dose that was high dose. So we're always comparing between high dose and standard dose. We never compared high dose to placebo. But in terms of blinding, I don't remember the exact number of how many studies were blinded off by hand, but I know that when we did the risk of bias calculations, about 5 of the studies had a high probability risk of bias. And so, but the rest of the studies were actually quite well conducted. I think it's the older studies that tended to be unblinded. And again, we think that that's where some of the bias could have been introduced.
Okay, thank you.
Okay, we still have a few minutes. Can I ask a question for Dr. Wang? I'm just interested. So your study was on HIV patients, and one of the recommendation now is to pursue this study, the 1 month. Yeah, the 1 month HP. So it's good if we're going to have a study for non-HIV population. But belonging to a low-income country like the Philippines, have you considered the difference in expenses of 1 month versus the 3 months?
Yes, thank you for this is a very good questions. I think I don't know which policy is best for Philippines or for other different countries. I think every country need to calculate its own cost and benefit for each policies. And in Taiwan, I think the most expensive part of preventive therapy would be the cost for DOT. The cost for DOPT, direct observed preventive therapy. So if we can successfully reduce the timing of dosing, I think that is the most money saving treatment. So I think in Taiwan, this is a very important consideration, is to bring a short course regimen, and as long as the side effect is acceptable by the population. Thank you.
Okay, thank you, Dr. Wang.
Yeah, could I ask a question about the age differentiation in the final presentation, Dr. Wang's presentation? And really the question relates to what are the hypotheses for the middle group, the group of the middle age range having the higher rates? Is it related to possibly alcohol, or is it related to some other thing? One would have assumed that as one ages, so the rate of adverse events increases linearly with or some relationship with age. But this was an unusual finding. Would you like to comment?
Yeah, thank you. Actually, it's very interesting for us too. And according from the preventive trial, the study had shown the highest SDR rate was occurred in the middle age group, but actually we cannot see. We also see the same phenomenon, and the SDR rate is less in the elder group. After review some studies, and we have some hypotheses. Maybe the middle age group that has a stressful life, and the body maybe under the low grade inflammation, and it might be stimulated easily after the drug stimulation. I'm not sure, maybe animal model or other cytokine data we can answer this question. And this study we are still going on, and it's very interesting indeed. Thank you.
Thank you very much. And then possibly a question to the second speaker, Buddy Susanto. I was wondering the change in time to positivity. If you could just comment on its, I suppose, its usefulness in determining the final efficacy measure of a TB agent. These changes seemed relatively small, I guess, but possibly you could just take us through some of the changes in the time to positive.
Okay, thank you for your question. So for the change time to positivity, it was based on an assay where we collect the sputum sample and grow the bacteria in the tube, and then we calculate the time until it gives the positive signal, until it reaches specific limit, and then it can how it change to positive. So the longer it gives to the positive signal, the less bacteria you will have. So I think it's kind of similar, but it's just using the time as the time endpoint instead of the number of the bacteria, I would say.
Okay, thank you. And there is a second question about the intolerabilities that you reported. And the question was related to hepatotoxicity. What proportion of the people who were unable to tolerate the higher dose, the 50 milligram, were there other adverse events apart from hepatotoxicity?
Yeah, there are some gastrointestinal side effects that are experienced by some of the patients. But yeah, I will. Yeah, it's quite many that has the most adverse effect are the gastrointestinal adverse event.
Okay, thank you. Elizabeth, over to you.
I guess that's it. No more questions. So maybe Neil, we can close the session now. We are on time.
I see there are two questions. One for Karen, I think, about what data would be required before 3HP could be used in pregnant women.
No, I saw that, and I actually wrote the answer in, but I can I can restate what I was going to say, because I think the person that was questioning it is also on the study to get more data. I think it is around that we know it's safe and tolerable to take in a pregnant woman. I think there is a need to have a larger powered study or larger studies to definitively characterize what safety is for pregnancy. And I think we do need the PK data for infants to understand if we do go forward with 3HP in pregnant women. So I know it's pending, and there's trials that are on the way under impact. So, and I believe they're going to be talked about at this conference and other sessions. So please carry that question forward, because we would support the need to get additional data to ensure that countries change the guidelines to include women who are pregnant on 3HP. Over.
Thanks very much. And then the final one that I see, and I apologize, Dr. Wang, again, from Gavin Churchyard, who's asking if you looked at the NAT2 genotype in terms of the adverse events. Was there any relationship between AEs and NAT2, or did you not look at this?
Thank you. That's also a very good question. I don't have the answer yet. We collected the samples, but I haven't the genotype of the NAT2. But in our previous studies, doing on close contacts who take the 3HP, we see that the side effect, the risk of systemic drug reactions is higher in patients with NAT2 slow acetylator. We confirmed our findings in both the TDM cohort and the SMP cohort. So at present, I think definitely the NAT2 genotype has something to do with the development of systemic drug reactions in 3HP regimen. Thank you.
Thank you very much. So I think it remains to thank everybody, particularly the presenters, for doing these presentations for us. And thank you very much to Elizabeth, my co-chair, for running this session.
Thank you to Neil.
And
so, just a reminder that, yeah. And I think this session can be viewed again on demand 1 hour after the session is finalized. Okay?